Welcome to the Deep Dive. Today we're diving into the very blueprint of life, DNA.
DNA is remarkable. Every tiny molecule packed with information.
It's like the ultimate instruction manual, right?
Yeah.
Telling our bodies how to build and run. But scientists didn't always know DNA was the key player.
Not at all. Early 20th century, scientists were focused on chromosomes.
Those thread-like structures.
Exactly. Inside a cell's nucleus. They knew they carried genetic information, but couldn't figure out if it was the DNA or the proteins inside that held the code.
A real cellular mystery. So what was the turning point? What pointed to DNA?
Bacteriophages.
Bacteria what?
Bacteriophages, tiny viruses that infect bacteria.
Okay.
They basically inject their genetic material like microscopic syringes.
Okay, microscopic syringes, I can picture that. But how do they help solve the DNA mystery?
Well, there was this brilliant experiment in 1952 by Alfred Hershey and Martha Chase.
Okay.
They tagged the protein and DNA in these viruses with different radioactive markers, like tiny tracking devices. They let the viruses infect bacteria and then tracked where the radioactivity went.
Like a stakeout, but at the cellular level.
Precisely. And the result? Crystal clear. Only the tagged DNA made its way inside the bacteria.
So is the DNA causing the infection?
Exactly, proving DNA was the carrier of the genetic code.
Wow. So DNA was the real hero, or maybe villain, in that cellular drama. But what is DNA made of? What are the pieces of this puzzle?
Imagine a long, twisted ladder.
Okay.
Each rung on that ladder is made of two molecules called nucleotides, and each nucleotide has three parts: a sugar, a phosphate group, and one of four bases:
Bases.
adenine, thymine, guanine, and cytosine.
Okay, I've heard of those. A, T, G, and C, right?
You got it.
So these are like the letters of the DNA alphabet.
Right.
Is there a specific order, or are they just randomly arranged?
Not random at all. Erwin Chargaff, another scientist, discovered something really interesting.
Well,
He found that the amount of adenine always equals the amount of thymine, and the amount of guanine always matches the amount of cytosines. Like A always fits with T and G with C.
like a puzzle.
Exactly. That's known as Chargaff's rule, a crucial clue to figuring out DNA's structure.
Interesting. So there's a specific pairing going on,
Yeah.
but how do they actually fit together to make this DNA ladder?
Now imagine the 1950s. The race is on to decipher the structure of DNA.
High stakes.
On one side, you have Rosalind Franklin, a brilliant scientist who used X-ray diffraction to capture images of DNA.
X-ray diffraction.
Yeah, it showed DNA had a helical shape. Think of it like taking a shadowy X-ray of a spiral staircase.
Okay.
So Franklin provides the first glimpse of this twisted structure. Now, enter James Watson and Francis Crick.
Ah, the famous Watson and Crick.
They were building models of DNA, trying to figure out the structure using all the data they had.
Uh-huh.
And when they saw Franklin's images, light bulb moment, they realized DNA is a double helix. Two strands of nucleotides twisting around each other, with the bases pairing up in the middle, A with T and G with C.
Just like Chargaff's rule predicted. Exactly. So Franklin's images were the missing piece.
Absolutely. They helped Watson and Crick build their famous double helix model.
I bet that was an exciting moment.
Yeah.
So we've talked about the structure, but how does this double helix actually work? How does it carry genetic information?
The beauty is in the sequence.
Mm-hmm.
The order of those bases, A, T, G, and C, along the DNA strand, that's the code.
The code for what?
It determines which proteins get made. And proteins are like the workhorses of the cell. They do pretty much everything.
So DNA is the blueprint, and proteins are the construction workers building and running the cellular city.
Exactly. And a segment of DNA that codes for a specific protein, that's a gene, like a recipe in a cookbook spelling out how to create a specific protein.
I see, it's all connected. The sequence of bases in the DNA determines the order of amino acids in a protein, and then the proteins carry out all the functions of life.
You got it.
But how does the cell actually read this DNA code and translate it into a protein?
That's where RNA comes in. RNA is like a messenger molecule. It carries the DNA's instructions out of the cell's nucleus to the ribosome.
The ribosomes.
That's where proteins are made.
So RNA is like a translator, taking the DNA language and turning it into something the ribosomes can understand.
Precisely. And this whole process, translating that DNA code into proteins, is what allows our bodies to function, grow, and even heal.
This is amazing. We've gone from a cellular mystery to the secrets of DNA structure and code.
It's a pretty incredible molecule.
It is. But I'm curious, what can we actually de-do with this knowledge?
Oh, that's where things get really exciting. Hold on tight, because next time on the deep dive, we're looking at how DNA is used to solve crimes, trace ancestry, and even protect endangered species.
No, that's what I call applied biology. Can't wait to dive back in.
Welcome back to the deep dive. Before we jump into the real world stuff, let's talk about ancestry. How scientists use DNA to trace it.
Oh, that's fascinating. I've always wondered how our DNA connects us to our ancestors, reveals our family's past.
Yeah. It's all about those unique patterns in our DNA, those genetic fingerprints we talked about.
Right.
Remember how some variations are more common in certain populations?
Yeah, like each population has its own set of genetic markers.
Exactly. So by analyzing those variations, scientists can actually trace migration patterns of humans throughout history.
Really?
It's like piecing together this massive global family tree, connecting people across time and continents.
So our DNA is like a record of our ancestors' journeys, like a hidden map in our genes.
It is, and it helps us understand not just where we come from, but also how different populations are related.
That's incredible. It's like having a historical record, but inside ourselves.
It really is.
But what about our health? Can DNA tell us if we're predisposed to certain diseases?
Absolutely. Some genetic variations can make you more susceptible to certain diseases.
Oh, wow.
So by studying these variations, scientists can identify potential health risks and maybe even figure out ways to prevent them or intervene early.
So it's like a personalized health report
Yeah,
hidden in our DNA.
you could say that.
That's both fascinating and a little scary.
It's true. Genetic information can be complex, and sometimes it raises difficult questions, but it also has the potential to empower people, to take control of their health, make informed decisions.
That's a good point. Knowledge is power, right?
Yeah.
Especially when it comes to our health. But let's switch gears for a second. You mentioned DNA technology being used to solve crimes. How does that work?
Remember those unique DNA fingerprints? They're incredibly useful in forensic science.
Oh, right.
By analyzing DNA samples from crime scenes, investigators can identify suspects, link them to evidence, or even clear someone who was wrongly accused.
It's like having a microscopic detective, right?
Exactly. DNA evidence has really revolutionized criminal justice. It's a powerful tool.
Powerful is right. And it's not just crimes, right? You mentioned paternity testing, too.
Right. DNA can help establish biological relationships between people.
Wow, the applications are really diverse. From tracing ancestry to solving crimes to understanding our health, it's amazing how much information is packed into DNA.
It is, and we've only just scratched the surface.
I can only imagine what the future holds for DNA research. It's pretty exciting.
It is. But before we get ahead of ourselves, let's recap what we've learned so far.
Okay.
We started by unraveling the structure of DNA, that double helix.
Right, with the base pairs.
Yes, forming those rungs of the ladder. Then we talked about how the sequence of those bases creates a code.
The code that tells the cell how to build proteins.
And those proteins are the workhorses of life.
And then we got into all the different ways DNA technology is being used. Ancestry tracing, criminal justice, healthcare.
Even conservation. It's really amazing how much information is in this one tiny molecule.
It is. One thing that strikes me is how DNA connects us to the past, to our history.
It really does. It sheds light on what makes us human.
Speaking of what makes us human, I'm curious about the ethical side of all this. DNA is powerful knowledge. It comes with responsibility, right?
Absolutely. As we learn more, we have to think carefully about the implications, how it can impact individuals and society. And that's a big part of our deep dive. We'll explore that in the next part of our episode.
Welcome back to the deep dive. We've been on quite a journey, haven't we? From the structure of DNA to all the amazing things we can do with it. But there's another side to this, right? The ethical questions.
Absolutely. As DNA technology keeps advancing, we have to think about the implications, for individuals and for society as a whole.
Like, we talked about how DNA can reveal predispositions for certain diseases. What if that information gets into the wrong hands? Could it be used to discriminate against people?
That's a valid concern. Imagine employers or insurance companies using genetic information to make decisions about who to hire or who to insure.
Wow. That would be a whole new level of discrimination.
It would. It's like judging a book by its cover, but on a genetic level.
So protecting genetic privacy is super important, right?
Yeah.
Especially as this technology becomes more common.
It's essential. We need regulations, safeguards, to make sure genetic information is used responsibly.
It's like having a right to keep your own blueprint private.
Exactly. And then there's gene editing. We talked about CRISPR a bit. Being able to actually change DNA with such precision.
Yeah, it's incredible, but also kind of scary.
Right. It has huge potential for treating genetic diseases, but there are also concerns about, well, unintended consequences.
We don't fully understand the genome yet, do we?
We're learning more all the time, but it's complex. Changing genes could have effects we don't anticipate.
It's like trying to edit a really complicated piece of software. Yeah.
A great analogy. One small change could mess up the whole system.
And then there's the whole designer baby thing, right?
Right.
If we can edit genes to cure diseases, what's stopping us from trying to enhance traits?
That's the slippery slope argument. It raises all sorts of questions about what it even means to be human.
It's like we're stepping into a science fiction novel, where we have the power to shape humanity's future.
It is a lot to consider. We need to have open and honest discussions about the ethics of gene editing. Scientists, ethicists, policymakers, the public, everyone needs to be involved.
It sounds like the ethical side of DNA technology is just as important as the scientific side.
I completely agree. It's not just about making scientific progress, it's about using that progress wisely.
Well, this has been an incredible deep dive. We've covered so much ground: the structure of DNA, how it works, the applications, the ethical questions. What a journey.
It has been fascinating, and it's a journey that's still unfolding. As we learn more about DNA, who knows what we'll discover?
And what new questions we'll have to answer. Well, thank you so much for being our guide on this exploration of DNA. It's been truly eye-opening.
It's my pleasure.
And for our listeners, remember, the world of DNA is full of wonder and complexity. Keep exploring, keep learning, and keep asking those important questions.